All right, hello everyone. It's going to be our last recording series on Chapter 17, Special Senses. So, yay! All right, so let's get started. All right, so we're going to be talking about smell, taste, vision, hearing, and equilibrium, right? So we may not necessarily think about it, but our sense of equilibrium is also going to be one of our special senses. And really, we do have other ones. Like proprioception is also considered a sense. However, it's not considered one of our special senses, right? In any case, again, smell, taste, vision, hearing, and equilibrium, they're housed in complex sensory organs. We'll talk about the different organs for each. And then just a couple of different definitions for you. When we talk about our sense of smell and our sense of taste, what we're really talking about is our chemical senses. That's what they're picking up on. They're picking up on chemical cues. Okay, I know we don't think of it that way, but that's what it basically is. So basically, our sense of smell and our sense of taste are interacting with molecules, right? So our receptor cells are able to pick up on very specific molecules, and then they send that signal to the brain, and that's when your brain interprets it as, whatever, banana, right, for example. Okay. And the other key thing is not only are these projections to the cerebral cortex, but they're also going to the limbic system for both. So in other words, your sense of smell and your sense of taste will evoke strong emotional reactions. And I think we all know that to be true, right? I mean, just think about it. I mean, no matter how many times someone may try to make, I don't know, a recipe that your mom or an aunt or a grandmother has made, it's never quite the same, right? And when you eat that pie, pasta, whatever it is, you're like, oh, it just takes you back to like when you were a kid or happy times or whatever, right? So these things have a very strong emotional reaction. And I think we've talked about sense of smell also having a very strong emotional component to it, right? You smell something, let's say it's a certain perfume or cologne, and if it reminds you of someone you dislike, you really get that visceral response to it, right? All right, I'm going to hide me here. All right, so we're going to talk about our sense of smell first. All right, so our receptors for smell are bipolar neurons. So if you kind of remember back to their shape, they're bipolar. They can be found in the nasal epithelium in the top of your nasal cavity. Okay, and if you remember back to when we did bones, if you remember the ethmoid bone, remember it had these little holes in the cribriform plate, right? Those little holes were for the axons of these bipolar neurons. Okay, so we're gonna see that these cells have supporting cells that help do just that. They help support the neurons, so they'll physically support them as well as nutritionally support them, as well as provide that mucous membrane lining the nose, keeping everything nice and moist. And then we'll have stem cells that produce new receptors. And these stem cells will replace these cells fairly frequently. We'll see the same thing in taste. I mean, if you think about it, these are receptors that are really encountering the environment. And if you smell something particularly caustic, and you might burn your nose, or hot might burn those receptors, they can replace themselves. Same thing with taste. Okay, and the other key thing here is that for both your sense of smell and your sense of taste, those chemical molecules will have to be dissolved in liquid. That's another reason why it's so important to keep these membranes moist. So the inside of your nose is moist, so is your mouth. If it wasn't, we wouldn't be able to taste or smell. Okay. So here you can see the olfactory epithelium here at the top of the nasal cavity. What you're seeing here are the nasal concha. If you remember back, here's that inferior middle that you saw in the skulls. And here's that superior one. It's way up here, so we actually didn't see that in the skulls at all, but it does exist. Okay, the actual epithelium right here is very small. It's about 1 square inch of membrane. But look at this. I mean, it is super dense with receptors. Anywhere from 10 to 100 million receptors in a square inch. That is a crazy lot, right? And again, you can see here's that top part is that cribriform plate of the ethmoid bone, and that's where those axons will be going through those olfactory foramina. Okay, so here's just a zoomed in image of that. So here you can see that cribriform plate. Here's that olfactory foramina, that hole. So here are the olfactory nerves, right? and here's this serous secretion, basically this liquid down here. You can see these odorant molecules, these little dots are dissolving in it, and if they touch the hair cells, or the hair part of the cells, the cilia, the receptor will actually fire and send that signal to the brain, right? And then here you can see, here's the olfactory bulb, so they impact on these olfactory neurons, and then again that sends a signal to the brain saying, oh, banana, oh, banana spray, whatever it is, oh, banana pie, okay? So right next to them in pink here are the supporting cells. These are columnar epithelium in shape. Again, they are there to help physically and nutritionally support the cells. And then you have these olfactory glands. This is what's actually producing the mucus, keeping everything nice and moist. Right? Oh, and then the basal cells, you can sort of see them here. They're smaller, maybe a little bit darker blue. These are the dividing cells that are going to replace those receptors. Right. So the way that this actually works is that the little odorant molecule is going to bind to the receptor, and by binding to the receptor, it causes sodium channels to open. And if you think back to when we talked about neurons and how they fire, how they depolarize, if we open up sodium channels, depolarization occurs, right? Because a positive charge is coming into the cell. Remember, we had the concentration gradients, right? So anyway, so odorants bind to the receptor, they open up sodium channels, you have depolarization, that nerve signal gets propagated down the axon to the brain. Right? We mentioned this when we talked about the different receptor types. So these olfactory receptors do show fairly rapid adaptation. So about half of your receptors have adapted in about a second. So that's definition of fast, really. And after about a minute, if you're in the constant smell, like there's no like wind wafting and then coming back, like if you're in a constant smell, in about a minute, you don't really smell whatever it is anymore. Give or take. These are sort of textbook numbers. Sometimes they could take a little bit longer, but again, not very long at all. Okay. In our sense of smell, I mean, it's not as great as a dog or anything like that. However, it's pretty dang good, right? It doesn't take a lot of odorant molecules for you to pick up on it. Look at that, 125 billionth, that's like decimal points, right? Billionth of a milligram per milliliter of air is all that is necessary for us to pick up on a smell. You tell this to that teenager who's covered themselves in Axe body spray. It's science. They do not need to bathe in this stuff, okay? Use this as a public service announcement. Anyway, so I think we all know people that wear way too much perfume, right? And cologne. It's like, oh, you don't have to. Our sense of smell is really sensitive. You can show them this slide if they need convincing. This thing here, methyl mercaptan. So I don't know how many of you guys actually have gas in your homes, but methyl mercaptan is actually actively added to gas so that we can actually smell it. So when you turn your stove on, or you turn whatever it is that you have fueled by natural gas on, you can actually smell the gas. And this is basically another thing that the government has decided to do as a public safety kind of thing. Back turn of the 1900s, early 1900s, they didn't have methyl mercaptan added to natural gas. So natural gas all by itself is odorless. We can't smell it. So what happened in places like New York City, where that actually things are predominantly gas there, is that people couldn't smell when they had a gas leak. So buildings would simply blow up because people didn't realize the gas was leaking. They lit a match, or lit a cigar or a cigarette or something, and the whole place blew up, and then obviously a big fire, right, would occur. And it still happens, right? It wasn't that long. It's been about 2 years now that a big apartment building in the north part of Manhattan actually blew up due to a gas leak. So again, methyl mercaptan added to natural gas as a warning. Okay. You don't necessarily need to worry about this. I'm not going to ask you the pathway. I just want you to understand how it works. So you go from the olfactory bulb to the olfactory nerve to the olfactory area in the either temporal or frontal lobes. There's no connection to the thalamus. This is a very primitive sense for us. So it makes direct connections to the brain. And again, also connects to the limbic system. There's another olfactory sense organ, and it's a little bit debatable whether or not humans actually have it. Some people say we do, some people say we don't. It's called the vomeronasal organ. So it's called that because it's near the vomer and near the nasal bone. So vomeronasal organ. Okay, you can kind of see it here in this cat. You can see it here in the snake. It's sort of basically, I guess, by the gums on the upper part of your maxilla. Okay. In any case, this vomeronasal organ detects pheromones, right? So when you see a snake stick its tongue out and it pulls it back in, what it's actually doing is it's taking the chemicals that it's picked up with its tongue and rubbing them along Jacobson's organ. When you see a horse doing this, what it's doing is it's trying to get the pheromones directly into that vomeronasal organ. And by the way, Jacobson's organ is the exact same thing as vomeronasal organ. Ever so often they just call it something, they call them either one. Names are interchangeable. Okay, same thing with the cat. Like when you see kind of like rubbing up against something and its kind of lip is up, what it's doing is it's trying to detect pheromones. Now, the reason why it's somewhat controversial in people is that we don't have an organ that's as obvious as in animals. It seems to be somewhat atrophied or vestigial kind of organ. However, in experiments, it has been shown that people can pick up on different pheromone levels in others. So they've done tons of different studies where they'll have, let's say, they'll have guys like work out in like a t-shirt, right? And then they'll present that t-shirt, that sweaty t-shirt, to a female. And depending where in her menstrual cycle she is, she will rate that t-shirt as really horribly stinky to, more pleasant. I don't think she'll ever say it's roses. However, it's not nearly as bad as, she gives it a more favorable rating, okay? And vice versa. They'll have a woman wear a different t-shirt each day of her menstrual cycle, and then they'll have guys sniff different t-shirts and ask them, which one smells the best, and they'll often say that it's the one that she wore when she was ovulating. So again, there is evidence that we can pick up on pheromone cues from one another. It's just that our vomeronasal organ is not as developed as animals, so there's still a little bit of back and forth as to whether or not it actually functions. All right, so let's just talk about our sense of taste then, because again, it's our other chemical taste, and in a lot of ways it's very similar and tied into our sense of smell. Okay, so again, just like in smell, the molecules have to be dissolved in order for us to pick up on them, right? We know that we have actually 5 different senses of taste. There's sour, sweet, bitter, salty, and a 5th one called umami. Okay, umami basically is the sense that gives us that savory or meat kind of depth to our food. Basically what this receptor is picking up on is monosodium glutamate. And the sense was discovered by Professor Kikuni Ikeda from Japan. And this MSG has gotten really a bad rap recently, well, within the last, 20, 30 years even. But really, all our foods actually contain some glutamate in them. So what I want you guys to do is take a look at this video on MSG. Okay? It's going to be very informative. And really, we're recognizing that MSG is not the bad molecule that, it has been demonized as being. So stop this video and click on this YouTube link and watch that and then come back. All right. So how is that video? Kind of interesting, right? So MSG isn't so bad after all. It's unfortunate how sometimes these things get stuck in the literature and no one really questions them. It becomes sort of like everyone just knows this, right? But there's nothing really to back it up. So maybe that's like a little bit of an example for you guys. When you hear something and you're kind of going, take that a little bit of extra time to find out exactly where that information is coming from. Okay. So we have the 4 classes of stimuli. There's sour, bitter, sweet, salty. And then basically any other taste, like, the complexity of banana, really is a combination of these plus umami. I should add in umami in here, plus our sense of smell. And again, you guys know this. You know when you're sick, right, and your nose is all stuffed up, everything tastes like cardboard. If you've ever eaten cardboard, you'd know what that tastes like. But it basically tastes like nothing, right? The right, you can't really taste anything. A really large portion of our sense of taste is actually smell. So if you can't smell it, you really can't taste whatever it is. And I think, we even use that as a trick. If you ever had to take that nasty liquid medicine that they say is cherry, but they're totally lying, right? You take that, you hold your breath, you just swallow it real fast so you don't have to taste it. Anyway, we do have quite a bit of taste buds, but look at this. I mean, we had 10 to 100 million in a square inch in our nose, a lot less taste buds or taste receptors in our mouth, right? So they're found on our tongue, on our soft palate, so that back portion of the roof of your mouth that's not bone, as well as the back of your throat. I think we've probably all had that really nasty, sour feeling like in the back of our throat when we're not feeling well or kind of nauseous, right? That's because you actually have taste buds back there. Okay. We have 4 different types of what are known as papilla. They're sort of these, structures on our tongue. So we have what are known as filiform papilla, fungiform papilla, valate or circumvallate papilla, depending on your textbook, and then folate papilla. Okay, so again, these 4 different structures on our tongue. Now, our taste buds are actually just located on the circumvallate, the fungiform, and then the folate. The filiform does not have any taste buds on it. Okay, so it does not have any taste receptors. If you actually look at this image in this filiform papilla, you'll notice they kind of almost look wavy, almost like, hair-like or something. They're predominantly there to increase friction on your tongue so that stuff doesn't just go kind of sliding all around in there. But you can actually move things to the back of your throat using your tongue. Folate papilla also, these tend to disappear too as you get older. So you probably had the sensation that your tastes have changed since you were a kid. Well, part of that is because you actually your taste buds will actually change as you get older. And one of the ways is that as you get older, you start losing some of the folate papilla. Okay, all right. So each one of these will have 3 different cell types. So the circumvallate, the fungiform papilla, and then the folate papilla, when they do have taste buds on them, those taste buds have 3 different cell types. So they have those supporting cells, the receptors, and the basal cells. So again, very similar to what we saw in the nose. All right. So here's a taste bud. Again, we have the hairs. We have the receptor cells with the hairs, we have the support cells, and we have the basal cells that are going to divide and replace things. So basically one taste bud has about 50 receptor cells making it up. There's a single hair cell from each receptor, goes up toward what's known as a taste pore, that's the opening. And very similarly, if there's a molecule that it is sensitive to, it'll pick up on that and then send that information. Oh, okay, this is sweet. Ah, this is salty, whatever. Okay. And these receptors we replace more often than we do our nasal ones. Again, that makes sense. I mean, just think about the things you eat and drink. How often have you burned the roof of your mouth or burned your tongue on something? A few days later, you're fine, right? That kind of burnt feeling is gone. Okay. So basically, very similarly, so the molecule will bind to the hairs, which will end up causing a release of neurotransmitter, and then that neurotransmitter will cause the postsynaptic neuron to fire, and then that signal gets taken to the brain. It has, I guess somewhat slow adaptation. So if you keep something in your mouth, and when we eat candy, right, you rarely leave it on one spot on your tongue, right? You kind of move it around your mouth. I mean, try this, at some point. Get like a piece of candy. It doesn't really matter, like a Jolly Rancher or something. Put it in your mouth, put it on your tongue, but don't move it. Like keep it on that exact same spot on your tongue for about 5 minutes. After a while, you won't actually taste it anymore. There'll be no signals coming from your tongue. I mean, you'll feel it. It's on there. But you won't taste the cherry or you won't taste the watermelon or whatever it is. Okay. Our most sensitive sense of taste is going to be bitter and that's because most poisons, like poisonous plants and stuff, taste bitter. So it doesn't. We respond to it very strongly. So in other words, it doesn't take much for us to pick up on bitter. However, we're less sensitive to salty and sweet, right? Things can have a lot of sugar in them. You could probably need a sugar cube, and that would be fine, right? Or a lot of, people just put salt on a lot of salt on their food. We can tolerate these in much higher doses than we could bitter. Okay, it doesn't take much. Right? And this also makes sense. I mean, just evolutionarily think about it. If it was sweet, had a lot of calories, you wanted to eat that. So yes, the sweeter the better, right? This is something called miracle fruit. This is awesome. Okay? So this little fruit is just absolutely fantastic. So what this does is if you eat the fruit, right, you kind of have it in your mouth, you kind of chew it around, you can even spit it out, you don't have to swallow it. What it does is it causes more sweet receptors to be inserted into your taste buds. So things that didn't taste sweet before because the threshold was too low, now all of a sudden they'll taste sweet to you, right? So for example, lemons, for example, actually contain quite a bit of sugar in them, but to us, they taste sour, right? If you were to chew on this fruit and inserted those extra sweet receptors in your tongue, the next time you tried to eat a lemon, it would actually taste like sweet lemonade to you. Crazy, right? At one point, they were back in the 60s, they were researching this fruit as a way to help diabetics. Because if diabetics, let's say like before they ate a meal, they popped like a little, some of these fruit or a pill made of these fruit in their mouth, you could actually give them basically sugar-free or very, very low sugar foods, and it would still taste sweet. So they wouldn't need artificial sweeteners and stuff that we know aren't good for you. What happened with that is the artificial sweetener industry was basically conducting research at the same time. At that point, it wasn't like the industry, it was basically one type. And they basically lobbied to have all the research done on miracle fruit stopped. However, don't let that stop you. You can actually get some of this miracle fruit tablets. They make them because this fruit is very sensitive, so after you pick it, it doesn't last very long. But they make them into tablets, and you can actually get them on Amazon. So I would say it's like $10 for like a pack. So I would say, try it out. Go get yourself a little packet, put it on your tongue. It takes about, 15, 20 minutes for it to dissolve, for the full effects to take place, and then try a lemon and see how that works. It's really cool. All right, anyway. Other things that can change our taste, which is really interesting, is hunger or starvation. You've probably all had this situation where you're really hungry and you go into a restaurant and you're like, I don't know, you get a burger or something. And you're like, oh man, it's like the world's best burger I've ever had in my entire life. It's like the best meal ever, right? So at some other point you decide to go back because this was such a good place to eat. You go back and you have her and you're like, oh, I don't know, why was I so crazy about this? This wasn't all that, and plate of fries, I don't know. But what happened was you were hungry, so your brain is like, no, you're gonna like anything. Anything that's edible, you are gonna like because we need to eat. So your brain will actually alter your perception of taste to keep you alive, which is insane, right? Another thing that our brain does without us being consciously aware of it. In fact, they'll have, when they talk to people who have been starved, and they'll eat like lots of weird things that we would never consider eating, like fish eyeballs and stuff. They'll say like, no, that stuff tasted great. I was actually craving it, which again is crazy, but that's a way that your brain does that to ensure that, yeah, no, I need the nutrients in that fish eyeball. It's going to taste good to you, so you eat it. Wild, right? Brain is so neat and like all the stuff it does. So that is really realizing it. So again, depending on your nutritional requirements, you may get different taste cravings. Certain things will taste amazing to you if you are lacking that nutrient. All right. So the way this works, so something that is salty, some salty foods, basically it's very simple. Salt, sodium is simply going to enter that receptor, and then basically you have sodium entering a cell. It's going to cause depolarization, neurotransmitters released, and you'll have an action potential in the postsynaptic cell, and that signal gets sent to the brain. In the case of sour, what you have is hydrogen ions actually entering the receptor. This should be a no there. Okay, basically, long story short, there's a bit more of a cascade that occurs. However, ion channels are going to open, eventually leading to depolarization and the release of a neurotransmitter, which then causes depolarization in the postsynaptic neuron, and that sends a signal to the brain. Now, sweet, bitter, and umami are a bit more complicated. You'll end up learning a little bit about G proteins in 202 if you haven't already. Let's just say this, it's a very long signaling cascade that occurs, but eventually you have the same sort of thing. There's depolarization that will occur, a neurotransmitter will be released, which causes depolarization in the postsynaptic cell, and then the information gets sent to the brain. Okay. Your sense of taste is actually conveyed by several different nerves. So you had learned about these in lab. So you have the facial nerve, the glossopharyngeal nerve, and the vagus nerve, and you can see each one takes up a slightly different portion of your mouth, as well as your palate, as well as the back of your throat. Again, you don't necessarily have to worry about this pathway. Just having it here for you, just so you know. Your sense of taste does go to the medulla, to the thalamus, and I should say and limbic system here, not or. As well as the hypothalamus, because remember that hypothalamus is going to be important in things like, hunger and stuff like that. As well as that primary taste center of the brain. So you can go, oh, okay, banana, instead of like, what did you just taste? Like, was it chicken? No, it's banana. That was close. No, you weren't. Anyway, so that's information going to the cerebral cortex gives you that conscious perception of taste. Like, yes, I have eaten banana. All right. So that's about that for smell and taste. So if you have any questions, as always, feel free to email me. Hopefully that all made sense, and I will see you next time.